Okay, the co-author first is from a PhD student and my colleague from Flinders University, and also we have two colleagues from US. Now, first, a little bit about the global context of this mangrove carbon sequestration. And this is a recent review paper. It summarizes all these anti-covariant measures, measurements on the wetland, including inland wetland and also coastal wetland. And the conclusion from that is the coastal wetland actually, the carbon sequestration strength is much larger than inland wetland. And then in the coastal wetland, the mangrove types of wetland, like intertidal forest wetland, is about 3 times the carbon sink strength is compared to average coastal wetland. And the other thing is we find is there's not many anti-covariant measurements based on these mangroves. So the red triangle that I show is globally only like 5 or 6 stations that published data shows. Now, this is our mangrove, Avicennia marina, right? So this is the only species that can grow in the latitude like in South Australia. And then with global warming, these species can spread further south. And so there's a potential to increase the coverage of these mangrove. And the potential significant to South Australia, and I used the average like 650 gram carbon from the global review study. The 650 gram carbon per square meter per year, and then use the South Australia's mangrove area, 156 square kilometer. So there you can get the carbon sink per year is 0.1 megaton per year. So this is about 1.2% of the South Australia's total greenhouse gas emission in 2015. So it's quite significant given that we only have 156 square kilometer mangrove. But to estimate this mangrove carbon sink strength is not that easy because there's big uncertainty. So this is a recent study actually study work on the same species that we have, but it's a bit short, only 2 meter high mangrove. And they only come to that about 74 grams compared to 650. So there's a lot of difference, 1 order of magnitude difference. So then the question is, how much carbon dioxide that mangrove in South Australia can sequestrate? And we actually, in November last year, we set up a station in St. Kilda mangrove trial. And you can see this photo. Then the inland side, right, is the mangrove is a bit short. I think it's probably under the stress of salinity. And the other side, go open to tide, that mangrove is quite tall, probably 6 meter high. And we set up the station in the middle. So this station, and when we set it up, our major purpose is try to test 2 systems. One system is a very common anti-covariant system, and the other system we set up is called maximum entropy production. That's why we have some collaboration with US. So if this method works, the maximum entropy production work, then we can reduce the cost from like 60K one per EC station to about 20K. So it can significantly reduce the cost. So that's the original idea. But now after some monitoring, we find that it's quite interesting to look into this mangrove carbon dioxide sequestration. So these are the data, 5-month data. The top panel show you is half-hourly data. And the bottom one is I integrate to daily. And you can see this is NE called net ecosystem exchange, is look at the surface as a sink or source of the atmospheric carbon dioxide. So if negative, it means it's a sink. So we like to see this negative value. So over these 5 months, So I average it, you get about 2.3 gram carbon per square meter per day. So if I use that, assume these other part of the year have similar strength, that they get to 850 gram carbon. So it's larger than global average, that average from that the global review study based on global measurement. So then I try to look at what factor could influence this mangrove carbon sequestration. So I plot this NEE versus daytime temperature. So remember that more negative is better, right? So it looked like we have a lower daytime temperature, actually mangrove fix more carbon dioxide. And then when your temperature get to like 32 degrees, and it doesn't fix any more carbon dioxide. That's the data show us. So then we think about why. So it probably, we could think at the higher temperature, you have more respiration, right? So then I plot the respiration based on nighttime. Nighttime NEE is a respiration. So, but although you see some relationship with temperature, but not that good. So then I try to subtract the respiration from net ecosystem exchange to get the gross primary production. So that tells you the production of the mangrove ecosystem in terms of fixed carbon dioxide. So in this case, positive is better because it fixes more productive. So better, and still is lower temperature is better, and higher temperature and get to the 30 degree, then it doesn't produce much carbon dioxide, fix much carbon dioxide. So here is our preliminary result based on 5-month data on the dry season in our area. So the mangrove in St. Kilda can fix carbon dioxide at the rate of 2.3 gram carbon per square meter per day. And then temperature appears to be the dominant control in these processes. But this is only very preliminary because I only get the data yesterday afternoon. So that's for sure. But if you have a bit more deeper thinking in this is the relation between these GPP, global primary production, and temperature. It maybe is an artifact of this how we measure the system, because this anti-covariate have a very big footprint. So that's, we call it source area. So you wind direction change, then you have a different source area. So in our high temperature days, usually we have a northerly wind, so its source area is here. And then colder day, temperature time, you have a southwesterly wind, so it's in this. So maybe that's temperature, apparent temperature influence. It could be because of this, the change of the wind direction. Now the other thing is based on that 2.3, right? That's based on dry season, our summertime. Now what about winter time? So this is the data based on global FluxNet data, and then you can see quite significant seasonal variation. So now the question to us is, what about this mangrove NEE in winter time? So we keep monitoring this. Now other thing we like to look at is about the salinity stress and also tidal activity impact on this mangrove carbon sequestration. And then one more thing is of this one. So you look at that about 5 or 6 kilometers away to our west. So we have a power generation plant. I assume they release carbon dioxide. And whether that carbon dioxide can be used sort of partly by this mangrove forest, that would be interesting, right? So perhaps if it can be used, that is good to know. Now, all these, so far, we just look at the carbon dioxide. We haven't looked at other greenhouse gas component, like, for example, methane. So these are sort of interesting questions we could further explore. I think with that, I'd like to acknowledge people. The project actually has not been formally funded by any organization so far. We use the existing equipment based on Flinders University, but we do got the support from City of Salisbury, Mark Purdy, he gave us very strong support for us to install the station. And then also the station so far is all intact, so it's in good condition. And also Jason Quinn from Duna also give us a support to set up the station. And Luke Mosley, and also he showed me one day the big mangrove. We try to set up in not the current station, but we found out it's very difficult. A lot of mosquito, right? Look, yeah. So. And then, although we are not funded by Goyder Institute, but the Goyder Institute, we have a project called From Soil to Carbon. It's just next to this site, so also provide some other connections so for us to set up the station. And a lot of colleagues from Flinders and students from Flinders give us help, particularly set up the station. I'd like to particularly mention Lawrence Berg, is a technician based in Flinders University. He used to be based at Adelaide University, so we're very good to have Lawrence to help us. Without him, probably the equipment still stay in our storage now. Okay, thank you very much.